Welcome to Moving Medicine Forward, a podcast presented by CTI Clinical Trial and Consulting Services. This podcast is centered on advancements in healthcare and elevating individual stories and their expertise. I'm your host, Jeremy Schran, and together with my co-host, Raina Graham,
hello,
we will take you through a deep dive of CTI, clinical research, the patient journey, and much more. On today's episode of Moving Medicine Forward, we will be discussing challenges in clinical research around two different types of cancers: acute myeloid leukemia, known as AML, and multiple myeloma, also known as MM. To aid the discussion, we brought in two of CTI's very best: Chad Jones, Senior Director of Clinical Project Management and Oncology Strategy Lead. He will be joined by Eric Clayton, Hematology Project Manager and Chair of the Oncology Center of Excellence. Let's jump right into the conversation.
Can you start us off by giving us a quick overview of AML and MM and why they are considered such difficult diseases to treat?
AML and multiple myeloma are basically characterized very simplistically as an excess of cells or a buildup of cells in the body. Multiple myeloma is characterized by an excess of plasma cells, and acute myeloid leukemia is characterized by an excess of blood cells. The direct cause of this, is not always perfectly well known, but what we do know is that they're extremely difficult to treat, and especially for AML, patients with this disease tend to progress much more rapidly than those with other blood disorders.
So, what are some of the key challenges when designing and conducting clinical trials for AML and MM treatments?
Yeah, so it really depends on what kind of trial you're trying to run and what kind of therapeutic you're trying to put in place. Obviously, if you're trying to run a CAR T study, that's going to come along with a lot of different logistics. The actual design of the study will vary because with a CAR T, you don't necessarily have the option of changing your dose as freely as you would with, say, a monoclonal antibody or some sort of an ADC or something like that. So it's really necessary for that design to be a certain style, and that usually is a 3+3 classic dose escalation design for those early phase studies. Whereas whenever you get into monoclonal antibodies or you get into small molecules, you're much more open to being able to fit the needs of Project Optimus, meaning that you're able to deploy or employ designs that are designed to get you to a very specific dose or better define a dose for your phase 2 studies and ultimately for manufacturing.
I can add something to that too, Chad. Just, talking about, a lot of the trials for AML and MM right now are, there's a lot of cell therapy. You mentioned CAR T, and the types of institutions that are comfortable performing cell therapy studies. A lot of them are our higher echelon, more famous, more experienced sites that we might work with. So those are often the sites that are most known to our sponsors and are sought after. So they're going to have lots of choices about which studies to run. So if you're trying to bring up a new study in this space, it might be difficult to find sites that would be interested in doing it because there's a lot of competition.
Yeah, Eric, you went into the site aspect of that. How does site selection and management impact the success of these trials? What are some of those current hurdles CROs face when recruiting qualified sites?
Yeah, so I mentioned you're going to go after the more popular, more well-respected sites, and they might have competition for the number of trials that or the number of sponsors that are interested in going to those sites. It might be hard to get your study into them. But the reason that we're more interested in seeking out those more experienced sites, they're going to have the personnel and the processes in place to manage these studies. A lot of your smaller oncology programs might not have the necessary things in place.
There are some very definitive things that sites that are conducting cell therapies, whether that be hematologic or that be solid tumor, have to have. And one of those is absolutely must have a cell lab. There is no way to get around that. If you're going to do autologous studies, you have to be able to perform apheresis. That can be done in an out center, but by and large, this is typically performed by the actual institution that is conducting the research. Now, for us, it is extremely important that there is sort of a movement to increase the number of sites that have the capability of doing this. And there's several reasons for that. One is to overcome competition. That is an absolute must. There are a lot of studies that are out there, and they're all vying for the same patient population. So The faster that we can move along with this, the faster that we're able to identify those patients that are outside those mega centers, those large academic centers. That improves our diversity largely, especially if we can engage in community centers that see a broader diversity of patients overall.
So CTI's team has a significant amount of experience with AML patients and cellular therapies. And we've been talking a lot about cellular therapies here. How does the CTI experience set us apart in supporting sponsors and improving trial outcomes?
CTI has been extremely fortunate to have multiple cell therapy trials, especially in hematologic malignancies, more specifically AML and multiple myeloma. From that's given us sort of an experience from the ground up. We started very, very early on in CAR-T. We saw some of the early developments. We recognized some of the barriers or holdbacks to that research, especially around the cells themselves, their ability to either shed antigen or to evade the therapeutic altogether. I think we've also learned over time that, especially for this specific indication, AML specifically, and multiple myeloma, that those cells do not express antigen equally across all of the cells that are being attacked. And so when the therapeutic is actually introduced to the patient, it may be extremely effective for certain cells, whereas others, it is just completely lost. It has no effect on them because they just don't express that target antigen. And as a result of that, those cells that are allowed to survive continue to proliferate and obviously are not beneficial to the patient.
So I wanted to talk more about the challenges with logistics. You talked about the importance of choosing specific sites, but why is it so important to get the chain of custody, that tracking and managing of the product from start to finish, out of the patient, back to the patient, and through that manufacturing? Why is it so important to get that piece of these trials right?
There's a couple of reasons for that. So you mentioned the chain of custody. So oftentimes for cell therapy study, they're going to have a leukapheresis at the beginning where we're taking healthy cells out of either a healthy donor or out of the patient. And then those cells will be sent off to a manufacturing facility where the cell product gets created, and then they'll be sent back to the treatment facility where they'll be put back into the patient. So making sure that all of that is running really smoothly can be incredibly challenging. We're coordinating with lots of different people and lots of different organizations. You have the site, you have the manufacturer, you have the courier service, and oftentimes, CTI as the project management organization is trying to coordinate with all these different people from all these different organizations, and we're doing a lot of that remotely. So just keeping tabs on everything that is going on. And make sure it's all running smoothly can be very, very challenging. One of the ways that we like to make sure everything will run smoothly is once we set this up, before we even have an active patient enrolled, we like to do a dry run just to make sure all the steps throughout this whole process are running like we would intend. And once we have that dry run complete, we can feel confident that everything is going to go smoothly when a patient actually shows up.
Eric, I'd just like to add a little bit of something there.
Sure.
The whole point of this is that we are able to treat the patient. And so getting this logistical pathway down pat, making sure that you have this practiced, very well understood by all the players that are involved in it, means that you're going to be able to move as efficiently and as fast as possible. AML patients, multiple myeloma patients, are quite often in a compromised state. By the time they make it to a cell therapy trial, they've had multiple treatments that have weakened them. Quite often, these patients are older patients. They may have comorbidities that are coming along with their AML as well. And they may have residual toxicities that are coming from those prior therapies. And so their time is limited. And again, AML can be quite progressive, quite quickly progressing whenever these patients have made it to this point in their treatment paradigm. So getting that whole process down correctly and making sure that nothing goes wrong truly can mean the difference between life and death for some of these patients.
Yeah, that's a good point, Chad. These patients are incredibly sick, and this whole chain of custody manufacturing process can take 4 weeks and take up to 6 weeks. And all the while we might be giving the patient some sort of bridging therapy or something to help them make it while this drug is getting manufactured for their treatment. But anytime we have an opportunity to shave a day or two off of that total process to get the patient treated more quickly, it's something that we want to make sure that we're doing.
So, depending on when you're listening to this podcast, the 66th American Society of Hematology, or ASH, annual meeting is coming up. So, what are you most looking forward to regarding research, emerging treatments, or advancements in hematology?
Yeah, Jeremy, I'll answer that. So I am going to have the opportunity to go to ASH this year. Every year, the excitement is around what has been accomplished in the previous year. So what's been accomplished in 2024, what worked, what did not work, and how can we then exploit those successes within the industry to improve therapeutics and get these drugs to our patients as quickly as we possibly can.
And if you're attending ASH and you want to meet Chad, we're going to be at booth 454. So stop by and say hi.
With these kind of unique treatments, are there any regulatory considerations that need to be in place, and how, as a CRO, do we help navigate those regulatory considerations?
Yeah, that's a good question. So the modification of cells in itself opens up a door for there to be higher scrutiny, right? And that you're taking a biologic and then you're reintroducing that biologic into a person. There is quite often a scrutiny around that. That's I think probably well deserved. How has the product been manufactured? What is the intent of that product, and how much benefit might that product actually have to the patient? I think this is all things that are going to be looked at by the FDA and regulatory bodies. Here at CTI, we do have a group that we call the RSA group that are experts in regulatory interactions. That is at the FDA level for the United States and other RAs around the globe. Whenever you come to internal bodies, let's talk about maybe IRBs or ethics committees within institutions. Our project managers had the opportunity to work with those institutions quite frequently. We kind of know what they're going to want to ask. For the United States, you quite often will see different responses from different either academic institutions or community centers. They may not always respond the same way, and they may not have the same committees or committee setups that you experience at others. So understanding that, knowing what the site landscape looks like is really important to be able to prepare your submission and make sure that you go through and get approval without there being much pushback.
How are CROs, pharmaceutical companies, and academic institutions collaborating to accelerate research and bring innovative treatments to market for AML and MM?
I think ASH is a prime example of that, Jeremy. We all come together once a year, discuss what our research has found, discuss what kind of successes or failures we've had, share that information either through publications or through posters, and also come together and have just open discussions about what we think the future of AML and multiple myeloma research will be. That will sort of determine where we go in 2025 for this year and each year moving forward.
So it sounds like all of these groups come together. We're, as a CRO, we're able to help sponsors anticipate those challenges and streamline operations to make sure these run smoothly. At ASH and just in general, what do you feel are the most unmet needs in this area of research, and how can the industry come together to fill those gaps?
I think this is probably the most blatantly obvious right now. The economic environment is a limiting factor for how fast we are able to bring these products to market. Funding has become somewhat scarce for many folks out there. The scrutiny around actual products and whether or not they look like they have promise and actually making it to market is being looked at such a level now that many promising investigational products are not receiving that funding, and they're not seeing the light of day, or at least they're going to be completely delayed or strongly delayed in their pathway to clinic.
Despite significant advancements in therapies for AML and MM, why do you think patient outcomes continue to decline at an alarming rate?
So I would not say that the outcomes are declining. I do think that we have made significant advancements in hematologic malignancies in general, but in AML and multiple myeloma. I think I may have mentioned this previously, but those two indications come with a host of difficulties in treating. One of those, again, is the shedding of antigen. So when you're trying to target a certain antigen, the cells develop resistance to that therapeutic very rapidly, and that makes it no longer effective. They also do not, they're not homogeneous, they are heterogeneous. So the antigens are not shared across all cells, and therefore some of those cells will evade and go on to continue to cause the patient problems. For some therapeutics like CAR T, which we feel is a very promising direction to go into, CAR Ts suffer from T cell fatigue. So a lot of times these patients enter the clinic, or they get to the point where they're about to participate in a cellular therapy of some sort that's utilizing T cells. And they have been through multiple treatments. They are very, very sick patients, and their T cells have just met an onslaught of attacks. So they are completely exhausted. So if you're taking these cells from the patient themselves, an autologous therapy, those cells are probably not in the best of conditions. And so as they are multiplied and prepared for that patient, or prepared to carry a marker of a certain antigen, those cells may not be in the best shape. And so they may not effectively attach themselves to malignant cells once they get back into the body. If they don't do that effectively, they're not going to do anything to the patient, or do anything for the patient.
Yeah, so you talked about patients being in a compromised state, and the importance of, obviously, patient retention is a huge part of clinical trials. Sometimes with the timelines and needing timely interventions, the delays in manufacturing can impact that. How does CTI mitigate the risks of losing patients throughout the trial, and how key is that in the CRO's role in that?
I mean, I think this comes back to logistics and making sure that your execution of the trial is absolutely perfect. There is nothing that we can do to improve manufacturing timelines. Once they come to us, once the sponsor comes to us with their product, they have already mapped out that manufacturing process, and presumably have perfected the manufacturing as much as they possibly can, which means they've shortened it as much as they possibly can. So they are completely reliant on us to get the logistics 100% correct, so that we don't carry any further delays.
Yeah, Chad, I'd agree with that. So, like, there's only so much in the entire time span between when there's a donation, or we get a patient showing up that needs treatment, to when we do the manufacturing and then get the cells back to the hospital where they can actually be treated. We can't really affect the manufacturing time point, like Chad said, but we can do things to speed up the courier process, or just maybe the way the schedule of manufacturing slots are made available to patients. We've had some studies in the past where there might only be a handful of manufacturing slots available in a given time period. So maybe the sponsor has made an arrangement to only have maybe 4 manufacturing slots over the course of a 6-month period. If one of the things that we will often suggest is that instead of having a limited number of manufacturing slots in a given time period, just make the manufacturing slots available whenever a patient shows up, because these patients are coming into the clinic, they're sick. We don't know when an eligible patient is going to show up. We can't predict that. So having everything set up and ready to go whenever a patient does present, that can eventually shave some time off the total manufacturing time.
So you talked about patients not knowing when they're going to show up. How does CTI deal with recruitment for these types of trials for patients that are critically ill like this?
So for AML and multiple myeloma patients at this stage in their treatment paradigm, they are very well known to the sites. They will already have a list of these patients that may be potentials for this. Obviously, they'll have to complete whatever therapeutic they are on at the moment. But once that's done, the investigator knows who that patient is, right? They know whether or not that patient is a potential candidate for the study. I think it's our job as a CRO to make sure that we are communicating with the investigator and site staff to make sure that they are looking at their list of patients and thinking about them critically, and whether or not they would be a good fit for this particular study, for whatever particular study they may be going on to. So quite often sites end up with an excessive amount of work. We understand that their resources become strained, and they may not have the time to do that patient search with diligence themselves. And so it's important that we are helping them through that, if as much as the site will allow us to. If they will allow us to do either screening logs, or they will allow us, our CRAs, when they're on site, to help the coordinators to actually identify those patients in their site, in their patient list, that is extremely helpful as well.
Another useful approach that I've had success with in the past, like you mentioned, helping sites run reports out of their systems, is these patients might be being treated for their cancer at these sites currently. So if we can run a report out of either their EMR or their pharmacy system for what treatments these patients would be prescribed to right now, that if eventually they should unfortunately fail that treatment, then they would become eligible for the study that we're working on. We could flag those patients with the site, and so they could be on their radar. And then if the patients do progress, then we know that they would be candidates for our trial.
So I think especially when it comes to cell therapies or in general targeted immunotherapies, I think there should be continued pressure to move some of these therapies forward in the patient's treatment paradigm. Again, many of these patients for both AML and multiple myeloma are older patients, and they are less tolerant to toxicities that you might experience through chemotherapies, which are generally provided in first line, or through some of the other even immunotherapies like checkpoint inhibitors and things of that nature. I think that some of these patients have a very limited runway as far as their treatments go. And the faster we are able to get what we feel to be the most effective therapies to that patient, the better off we will be, even though it sort of defies the standard of care these days.
Thank you both for being on the podcast today. We're so lucky to have this expertise in our company, and thank you so much for bringing your expertise to the podcast. And speaking of that expertise, we are always adding to the team. CTI is currently recruiting a senior medical director in hematology and oncology to enhance our expertise in these critical fields. So if you want to learn more, you can come meet us at booth 454. Dr. Monica Shaw, our new chief medical officer, is going to be there as well. So you can come meet her and see what it's like to be on the team. If you want to find out more about that job, you can always go look at our website at ctifax.com and then /careers, and it'll take you to the careers page where you can go look at all the great opportunities we have here. I'm sure Chad and Eric, you would say that it would be great to join the team, and you get to go work with you guys.
Right, absolutely. Whenever I started at CTI, I was extremely impressed with the complexity of work that CTI had done. So we typically engage in studies that are as I said, complex, but are also groundbreaking, very much so novel, the sort of thing that investigators typically find to be very, very interesting and promising as far as treatment goes for their patients. This is not just for AML and multiple myeloma. I think for all hematologic disorders, even outside of oncology, we have just an amazing breadth of experience that you can draw from for just learning from, but also add to, and also know that that's the driving force for us. We are going for these complex trials. We're going for these potentially treatment-shattering studies that really get the attention of investigators.
I would agree with that. That's one of the things that I like about working at CTI. Chad, you know that I'm involved in a lot of the phase 1 dose finding first in human studies, and these are the novel types of therapies. These are some of the first times that these therapies are being tried in human participants. So just seeing that innovative type of experimentation is one of the things that I find really interesting. And I think a lot of people like being a part of.
And I think it's worth noting, Chad, you oversee it. And I know, Eric, you've contributed to a lot of components of our Hematology Oncology Center of Excellence. And that's really CTI trying to find innovative ways to combine all the knowledge and progress these trials forward so that we can impact medical advancements and move medicine forward to help these critically ill patient populations.
Yeah, thanks for having me on the podcast today. I look forward to seeing everyone at ASH 2024.
Thanks for having me on the podcast.
Thank you for listening. If you enjoy discovering how medicine is moving forward, or you are interested in how CTI could help you with clinical research, be sure to subscribe. Follow us on social media or visit our website at ctifax.com.